Hi, my name is Jim Carr, Chief Clinical Development Officer for Stealth BioTherapeutics, and I'm going to talk about lessons learned from the SPIM 301 program. To start out with, and I'll talk about some of the lessons as we go through the presentation, but these are the top line lessons learned. What we've learned about ilimipritide is that nuclear transcription and protein importation appear to be important parts of the mechanism for the drug. We've learned that longer duration of therapy with ilimipritide appears to be important for the treatment of chronic diseases. What we've learned about the trial design is that the basket trial design introduced just too much heterogeneity to succeed in this patient population. We learned that a lack of biomarkers presented challenges from the beginning of our efforts in this disease area. A lack of longitudinal natural history data presented challenges. For PMM, we've learned that there remains a high unmet need in this patient population, particularly with respect to weakness and fatigue during activities. We have learned that objective, reliable, and validated endpoints to measure muscle weakness and fatigue are needed. And we've learned that there are highly dedicated investigators, site staff, patient advocacy, and patients, which allowed the successful completion of this developmental initiative. Just a little bit about the mechanism of the drug. With normal mitochondria, as pictured on the left, the role of cardiolipin is to hold the electron complexes together. This allows essentially this efficient electron flow between the complexes. When this happens, there's generation of ATP. There's a balance of oxidative stress and free radicals. This permits protein importation to occur, supercomplexes to form, and there's a balance of fission, fusion, and mitophagy. In disease mitochondria, the cardiolipin molecules can become peroxidized. This allows the complexes to essentially drift apart, and this disrupts the normal flow of electrons. As a result of that, there's a diminishment in ATP, there's an increase in oxidative stress and free radicals, there's a reduction in protein importation, supercomplex formation, and there's impairment in the fission, fusion, mitophagy process. And we believe that in particular, the top three aspects of this are really important for the mechanism of action of the drug. Just as a reminder, ilimipritide is a 4-amino acid peptide that associates with cardiolipin in a 1 ilimipritide to 2 cardiolipin molar ratio, which improves mitochondrial structure and function, which we've seen in numerous disease models. So for the protocol design, methods, and safety, this was a trial that was conducted in 7 countries at 27 sites. 296 patients were screened to get to 218 patients randomized. Some key inclusion/exclusion criteria are that the patients had to be 16 to 80 years of age. Patients had to have a genetically confirmed diagnosis of primary mitochondrial disease, and they had to walk greater than 100 and less than 450 meters at both screening and baseline visits. Patients were then randomized to drug or placebo and treated for 24 weeks. Upon completion of the trial, the patients could roll over into the open label extension. The primary endpoints were a change from baseline in the 6-minute walk distance at 24 weeks and a change in the total fatigue score, also known as the PMMSA, at 24 weeks. The patients were equally matched, 109 patients in each group, 218 total. Just a little bit about the analysis populations. So ITT, all subjects are randomized appear in the intention to treat population. The safety population likewise is 100% of all subjects. The per protocol population is really comprised of subjects that really finished part 1 of the study. So the patients completed all assessments and they took study drug very compliantly through the course of the study. So there were 7 subjects that did not complete part 1 in the ilimipritide group and 6 in placebo. So I will be talking about a per protocol population, so I wanted to go through the definitions of that population. And those populations were decided before the blind was broken on the study. The baseline characteristics, predominantly a white population, about 2/3 female, which is consistent with our phase 2 effort. To me, one of the most noteworthy findings at baseline is the really low 6-minute walk test performance. So these are essentially 45-year-old subjects walking 330 meters at baseline. And that's a really poor performance in the 6-minute walk test. That's similar to moderate symptoms of heart failure, which is known to cause profound exercise intolerance. So these patients have a lot of evidence of exercise intolerance at baseline. The total fatigue score is modest. This was a 4 to 16 scale, coming in at 10.6, which was a bit lower than we saw in phase 2. We actually used a stratification scheme. So this was pre-specified to make sure that we had balanced groups with both mitochondrial DNA and nuclear DNA mutations. And we used a de Morrow classification, and this is how the patients are categorized. I've boxed the nuclears just to call attention to the fact that we have studied the drug in another nuclear population, and that would be Barr syndrome, and that is also part of the de Morrow classification, and it's under alterations of the lipid milieu. So as I talked about earlier, about 50% of patients listed tiredness during activities and muscle weakness during activities. Those are most bothersome symptom. So the learning is that there remains a high unmet need in this patient population, particularly regarding weakness and fatigue during activities. This is the AE profile. Not a surprise, most of the AEs were comprised of evidence of injection site reactions. For example, injection site erythema or redness at the injection site was experienced in about 86% of subjects and 28% of placebo, but most of the adverse events are consistent with injection site reactions. For efficacy, so for the intent to treat population, and this was the finding in the 6-minute walk test. So you can see that there was evidence of an improvement in both groups at week 4, then again at week 12. And by week 24, the improvement in the placebo group surpassed the effect in the elimipritide group. So it was about a 3.2 meter improvement greater in the placebo group. That was not significant. So the lesson learned is that the placebo response in the 6-minute walk test underscores the need for additional objective, reliable, and validated endpoints to measure muscle weakness in this patient population. And this is the finding of the per protocol analysis. It's really no different than what we saw with the intention to treat population. This is the finding on the other endpoint. This is the PMMSA, the total fatigue score. Again, evidence of a reduction, which is a desirable movement in this endpoint. So a decline was observed in both elimipritide and the placebo-treated patients. At week 24, a bit more of an improvement in elimipritide, but it was a small change. So the lesson learned is that moderate fatigue at baseline underscores the need for additional objective, reliable, and validated endpoints to measure fatigue during activities in this patient population. Here are the findings with the per protocol analysis. Again, it doesn't change the overall assessment of this endpoint. This is the performance by subgroup on the 6-minute walk test. So on the left part of the slide is the subgroup comprised of the mitochondrial DNA mutation subjects. Again, this represents about 3/4 of the trial population. So as you can see, similar performance in the 6-minute walk test at week 4 and again at week 12. And by week 24, there was about an 11-meter improvement in the placebo group that was greater than the effect observed in the elimipritide treated group. Contrast that to the findings of the nuclear DNA mutations. So two things that stand out in this slide. First of all, the placebo group, there's really no evidence of increase. So there's really no placebo effect that was observed in the nuclear DNA genotype subgroup. At week 4, there was about a 23-meter improvement in the elimipritide treated group. And by week 4, it was still a 16-meter improvement. So the difference from placebo was about a 16-meter difference. So the lesson learned is that the basket trial design just introduced too much heterogeneity to succeed in the overall population. Now the per protocol population here, again, this is really looking at patients that completed the trial. So again, not much increase in the placebo group. So really little evidence of a placebo effect in the placebo-treated patients. And at the end of treatment, it's about a 24-meter difference between placebo and elimipritide treated patients. and that was nominally significant at 0.05. The lesson learned is that nuclear transcription and protein importation may be important for the elimipretide mechanism of action. Also, the lack of placebo response may indicate a reduction in mitochondrial reserve capacity in subjects with nuclear DNA mutations. For the nuclear subgroup, this was the majority of these subjects had a POLG mutation, and followed by twinkle, but it was the majority of the subjects had a POLG mutation. These proteins are translocated into the mitochondria through translocase outer membrane complex, which requires cardiolipin for its structural integrity. So again, we think that's an important finding pertaining to the elimipretide mechanism of action. So just a couple of comments about Barr syndrome, which again is a nuclear mutation which causes alterations of the intermitochondrial membrane lipid milieu. Barr syndrome, you might know, is characterized by a cardiomyopathy, skeletal muscle weakness, neutropenia, and premature death in these subjects. What we've learned is that a longer duration of therapy, greater than 1 year of exposure, resulted in greater improvements from baseline in open label extension. So even week 36 represents a combined 1 year of therapy. So we've seen really noteworthy improvements on 6-minute walk test, muscle strength. We've seen reductions in fatigue. So what we've observed in Barth is that a longer treatment duration may be necessary. So the lesson learned is longer duration of elimipretide therapy is important for chronic disease treatment. For Barr syndrome, we also benefited from pretty rich natural history. So even though it's a really small trial population, the availability of natural history data facilitated the trial design. It really allowed us to choose endpoints, for example, that were meaningful in this patient population, and also enabled a retrospective natural history control analysis to occur to really benchmark the findings that we were observing in open label extension. So you can see in the 6-minute walk test and the handheld dynamometry, there's really little improvement that occurs naturally based on the natural history. Likewise with the 5-time sit to stand, and we can compare the effect with elimipretide to what would occur naturally. So again, it just allows us to benchmark the findings to our open label extension data. So the lesson learned for PMM is that longitudinal natural history data would be really helpful. And the lack of it really presented some challenges in the design of our trials. Another comment about Barr syndrome, all of the subjects in our trial had evidence of cardiac impairment at baseline. So what we observed is that the drug led to an improvement in heart function as evidenced by stroke volume. What this allows for with the clinical trial is that this can really be viewed as a biomarker. So if we had an objective finding like this in PMM, it just allows you to essentially assess whether or not you're getting target engagement, for example. And it also permits, for example, an enriched design. So you can actually target patients that have responded to the drug. So in summary, lessons learned, just to recap, lessons about elimipretide. So we've learned that nuclear transcription and protein importation with elimipretide stabilizes cardiolipin, which among its other functions facilitates the transcription and importation of the nuclear proteins. We learned the observation of an improvement in the 6-minute walk test in subjects with nuclear DNA mutations may indicate that nuclear protein transcription and importation is an important aspect of the mechanism of action of elimipretide. The SPIM301 nuclear DNA response is consistent with the observations in Barth, that is, a nuclear DNA defect in a homogeneous population. For duration of therapy, we've learned that a longer duration of elimipretide therapy may be necessary for chronic diseases. For the trial design, we learned that a basket design just added too much heterogeneity between the mitochondrial and nuclear DNA mutations, which was particularly apparent with the 6-minute walk test results. The observation that placebo-treated subjects with nuclear DNA mutations were unable to increase their exercise performance may indicate a lack of mitochondrial reserve capacity in this population. For biomarkers, effort-dependent and subjective endpoints to assess neuromuscular symptoms in PMM introduce risk in a heterogeneous patient population with differential mitochondrial reserve capacity. A biomarker-targeted approach, such as observed in Barr syndrome with improvements in left ventricular function, could provide assurances of target engagement and support enrichment strategies. For natural history data, the pretrial registry, also known as SPIN 300, added to our knowledge of natural history, but did not provide sufficient long-term longitudinal data to de-risk effort-dependent endpoints. Lessons about PMM, the unmet need, tiredness during activities and muscle weakness during activities were described as the most bothersome symptoms for 50% of the subjects, and this remains an unmet need. Objective, reliable, and validated endpoints to measure muscle weakness and fatigue are needed. For the conduct of the trial, the pretrial registry, again, SPIN 300, greatly enhanced recruitment. The dedication of the investigators and associated study staff allowed successful execution of the trial. And the commitment of the patient community and advocacy to foster therapeutic development facilitated key aspects of the development program, including trial enrollment, PRO development, and human factor studies. As has been the case in other therapeutic areas, phase 2 results can be misleading, which reinforces the importance of objective biomarkers to guide and de-risk drug development initiatives. Thank you for your attention.
Hi, my name is Colin Meyer. I'm the Chief Medical Officer at Reata Pharmaceuticals, and I'm going to talk about lessons learned from the MOTOR study, which was a study in patients with primary mitochondrial myopathy. Standard disclosure slide. And so I'm going to talk about the rationale for use of omavaloxolone, or OMAV, in mitochondrial myopathy, the design of our MOTOR trial, the biological results from the MOTOR trial, the clinical results from the MOTOR trial, and then the results from the MOXIE study in patients with Friedreich's ataxia, and then conclusions and lessons learned from the MOTOR trial. We've been working with our drug OMAV for some time to understand how it works in cells, animals, and people. And researchers have shown that our drug, which activates an important regulator of energy in the cell called NRF2, that regulator directly affects the cell's ability to produce energy. In work that was published by academics, they demonstrated that when the target of our drug, NRF2, is suppressed, animals have impaired energy metabolism and they have impaired exercise intolerance. Conversely, when the target of the drug, NRF2, is activated genetically in animals, that results in more efficient use of stored fuels, such as glucose and fats, that results in increases in energy production and increased ability to walk in animals. On the basis of those data and a much larger data set demonstrating that OMAV can restore energy production in biopsy samples from patients with mitochondrial disease, we developed the MOTOR study. The types of patients who are enrolled in the MOTOR study are shown here. Patients had to have a mutation in either nuclear or mitochondrial DNA that's associated with mitochondrial disease. Patients had to have a history of exercise intolerance. They had to be able to perform exercise on a recumbent bike so we could measure physiological and clinical parameters. And they had to have impaired exercise capacity as defined by peak work of less than 1.5 watts per kilogram. In the trial, we enrolled a variety of patients with various types of clinical diagnoses. But once again, all these patients shared in common mitochondrial disease and impaired exercise capacity. The trial was 12 weeks in duration, and patients were given either OMAV at various doses or placebo. Patients, investigators were blinded to study medication, so they did not know what they were receiving. As you can see here, we had progressive cohorts starting at a low dose of 5 milligrams and worked up to 160 milligrams. Patients, once again, were treated for 12 weeks, and they had multiple assessments during that. So during the baseline period before patients received drug or placebo, they had a series of tests that I'll describe on the next slide. And these were again conducted at weeks 4 and then at the end of treatment at week 12. There were three major types of tests that were conducted in the motor study. We wanted to understand if our drug could induce NRF2, the target, in patients with mitochondrial disease. We wanted to understand if this would translate to improvements in physiological parameters. So you can see on the left, patients had a test called the submaximal exercise test, where they pedaled at a constant rate for 30 minutes. And during this test, blood was collected to assess for lactate levels, and then the heart rate was assessed. And when patients deplete their aerobic capacity, heart rate goes up, and blood lactate levels go up as well. And so we wanted to determine if our drug could show separation and increases in heart rate and blood lactate levels versus placebo. The primary endpoint of the study was a clinical measure of the peak work as determined on an exercise bike. And so patients would start at relatively low resistance in every 1 to 2 minutes. The resistance would increase, and therefore the amount of work required to pedal would increase until patients reached the maximum amount of work that they could conduct. And the third major test that we did is called the 6-minute walk test. And so this is just as it sounds. Patients would walk for 6 minutes, and the total distance that they walked is a surrogate for how patients are doing clinically. We were able to show in patients' blood that NRF2, the target of OMAV, was induced. And so you can see the top panel here, the successive increases in bars at increasing dose levels, demonstrating that the drug was dose-dependently inducing the target NRF2 in patients with mitochondrial disease, demonstrating that it was biologically active. And you can see in the bottom panels that our drug, when compared to placebo, was reducing blood lactate levels and reducing heart rate. This was both dose-dependent, and so it improved at increased doses relative to placebo, and it was time-dependent. And so the changes at week 12 were greater than the changes at week 4. So from our perspective, we were able to show that the drug was in fact biologically active in patients, and we were able to demonstrate the drug was having a physiological effect and was in fact improving mitochondrial function in patients using these blood-based and physiological assessments. From a clinical perspective, we saw no change in peak work. You can see the graph here. There's like no difference at placebo or all doses or the optimal dose of 160 milligrams. So we didn't see a change. The test could have been insensitive to change to our drug, or our drug may not have been able to affect it, but there was no change observed at any dose level. You can see the bars on the bottom for the 6-minute walk test. This is the median shown. And so you can see that patients across all dose levels showed a very slight increase versus placebo. And then the optimal dose, there was no change. One important observation from this assessment was that there was a lot of variability. And so patients appeared to be able to behave differently at different time points. And the most notable observation was that in the placebo group, we saw high variability. And so while theoretically a patient who is receiving placebo and who has a chronic disease should progress at a slow rate and really not change across a 12-week treatment period, there were 2 placebo patients who had increases of more than 100 meters, which is a very large change. In other diseases where 6-minute walk distance has served as the basis for approval, changes as low as 10 to 11 meters have been considered clinically meaningful and have been served as the basis of approval. And so the fact that 2 placebo patients had large increases just demonstrates how variable this assessment is in this patient population. For comparison, we were conducting a parallel trial in a separate set of patients who have a disease called Friedreich's ataxia. This is also a mitochondrial disease, but this trial only studied this set of patients. Instead of these patients having severe fatigue and exercise intolerance, these patients primarily have issues with coordination of their muscles. And so this study, we also assessed for biological activity of NRF2 targets. And so I showed you previously in the motor trial, we saw improvements in GGT. You can see in the panel on the right, in both studies, we saw similar improvements at a given dose. We also looked at another marker of NRF2 induction called ferritin, and we saw similar regulation. And so to us, this demonstrated that OMAV was also biologically active in patients with Friedreich's ataxia to a similar level that it was in patients with mitochondrial disease. One difference in patients with FA versus patients with mitochondrial myopathy is that there has been an extensive set of patients who've been studied to help validate a sensitive clinical outcome measure. And so the MFARS, or Modified Friedreich's Ataxia Rating Scale, is a physician-assessed neurological exam that tracks progression of FA. This was developed in collaboration with FARA, which is the main patient advocacy group, and the FDA. It has 4 sections that are considered clinically meaningful. And so they're shown here in the top right. One of them measures the ability for patients to speak and swallow. One section, upper limb, measures coordination of patient's upper limb. Another section measures coordination of the lower limb. And the last section measures patient's ability to stand and walk, which is the upright stability section. FARA has enrolled a natural history study that currently has enrolled over 1,000 patients, and they have follow-up on MFARS for up to 10 years, and they have several key publications. The one on the bottom includes data for approximately 500 patients for 5 years of follow-up. And notably, they've been able to show that the MFARS score worsens over time at 1 to 2 points, and this has been characterized, and this has been linked to important clinical milestones in patients' disease, including the average score when patients are diagnosed, the score at which patients lose the ability to walk, and the score at which patients typically die. There's been a lot of dialogue with the FDA about MFARS, and they've publicly stated to us and to other sponsors that MFARS is an approvable endpoint in Friedreich's ataxia. What we demonstrated is shown here. The pivotal portion of this trial was 48 weeks in duration, and so 4 times longer than the motor trial. We did do a dose ranging portion in the motor trial that was 12 weeks, but this is the definitive efficacy portion of the trial. We randomized 82 patients to OMAV or placebo, and you can see here there was an improvement or reduction in score in both groups at week 12, which isn't surprising since there can be a placebo effect in many trials where there's an effort dependency to the assessment. But you can see that the curve separated so that by the end of 48 weeks, there was a separation of 2.4 points relative to placebo, which is roughly equivalent to 1 year of progression of MFARS. And this result statistically was significant with a p-value of 0.014, meaning it was very unlikely due to chance. We measured other assessments in this trial, including instruments to determine if patients reported if they improved, and there were numerical improvements in that, whereby more patients who received OMAV said that they had an improvement in clinical status versus placebo. And we showed that this correlated with the physician assessment of their status, or MFARS. And we also conducted a questionnaire for activities of daily living that measured many important aspects of patients' daily lives, including their ability to swallow, cut food, dress, and walk. And that showed nominal significance as well. And so in conclusion, to support approval, a drug must demonstrate an improvement in a way that a patient feels, functions, or survives. And there's very specific legal requirements and definitions of what an endpoint can be. And so it's very important to develop, validate, and establish objective clinical endpoints that can be used in mitochondrial disease trials. In the motor trial, we very clearly showed biological activity as well as improvements in physiological parameters that to us demonstrate that the drug is active in patients and was improving mitochondrial function. But unfortunately, we did not see any improvements in clinically meaningful outcomes measures that could serve as a basis for approval. Conversely, in the trial in Friedreich's ataxia, we did see an improvement in biological parameters that also translated to improvements in clinical assessments. So from my perspective, it's important to continue to determine appropriate, sensitive, and validated measures of disease progression that can then be used in clinical trials, which would further facilitate drug development in mitochondrial diseases. And from a trial design perspective, I think there's multiple lessons learned. Obviously, the first, as I've been discussing, it's important to have an appropriate clinical endpoint that you hypothesize your drug can affect. It's important to minimize variability so you can show a difference. You don't want the trial to be noisy, and so selection of patients into a trial has to be important to identify the right patients. And then thirdly, you must have sufficient number of patients in a trial to show statistical separation. A drug may clearly work, but if you don't have enough patients in the trial, you may not be able to show that from a mathematical and statistical perspective that the drug is working. And that has been a challenge in mitochondrial disease studies because there's so few patients with disease. And then lastly, I think a clear lesson from the Friedreich's ataxia trial is that the trial must be long enough to account for any placebo effect or improvement within the first few months. That effect needs to wash out. And then furthermore, and ideally, there needs to be separation. And so there has to be time for patients on placebo to have some amount of disease progression so that if a drug is working, a significant separation can be shown. And so I think insights from all the trials that have been conducted in mitochondrial diseases, those that have been successful and those that have been unsuccessful, really do increase our understanding of what needs to be done in order to design more efficient trials in the future so that hopefully we can find treatments for patients with this disease. Thank you very much.
Thank you very much. I'm excited to share with you all today lessons that we've learned from the development of mitochondrial disease drugs over the past decade. The purpose of this presentation is to share the key learnings that we've collected from the conduct of controlled clinical trials worldwide in patients with primary mitochondrial disease. First, I just want to go over the ideal attributes for drug development. In general, when you think about initiating a drug development program, you think about developing drugs for a disease that is homogeneous, where there's a shared genotype, phenotype, constellation of symptoms, and rate of progression. It's also important to have a natural history, so that the natural history of the disease is well understood from its onset to its end stage, including rate of progression. Having objective metrics of disease that are easily measured and correlate with disease severity are also critically important, as is having an accessible population where there's a sufficient number of patients who meet entrance criteria for the trial and can participate in clinical trials. And finally, it's very helpful for there to be a precedent for drug approval so that there's a clear and established development path. Now, as we think about mitochondrial disease, it's very clear that mitochondrial disease does not have most of these attributes, which raises a number of challenges for disease development. In addition, this concept that there's no previous approved therapies becomes really, really important when we think about initiating a drug development plan, as there's no established pathway forward, no validated endpoints, no model for clinical trial design, and oftentimes there's a limited culture of clinical trial participation, and we'll find that there's a lot of education needed for patients and investigators, particularly around the concept that a clinical trial is not clinical care. Now, this was certainly the case when we began our work in mitochondrial drug disease development over a decade ago, and I'm proud to report that many of these issues have gotten better, yet they still remain challenges. As I think about the key lessons that we've learned over the past decade, here are the top 7. 1, don't confuse rare with homogeneous. 2, accurate and comprehensive natural history is critical. 3, biomarkers are needed to facilitate all stages of development. 4, objective metrics of disease are better endpoints than composite scales. 5, short trials lead to long delays. 6, an aggregated patient community is critical to patient recruitment. 7, challenges exacerbated by disease rarity and barriers to travel to study sites. So let me run through each of these one by one. First, lesson 1, don't confuse rare with homogeneous. Mitochondrial disease has enormous heterogeneity, from a genetic standpoint as well as a phenotypic standpoint. Disease can arise from mitochondrial DNA mutations, nuclear DNA mutations, and there's also the issue of heteroplasmy. In terms of phenotypic heterogeneity, you can have the same genotype, but different disease course within that specific genotype. In addition, different organ systems can be affected, severity of symptoms can vary, and disease rate of progression can vary, again, even within the same disease subtype. Next, lesson 2, accurate and comprehensive natural history is critical. When you think about initiating a drug development program, having a clear understanding of the disease course is incredibly important. And it turns out that just simple anecdotal or case series don't really provide the true natural history of disease. In addition, it's important to have an understanding of disease progression in the context of optimal care. That is, understanding what is the progression of disease when patients are getting the appropriate level of medical care, physical therapy, and nutrition support. That helps us understand what a disease progression could be like in a clinical trial. The absence of a natural history prevents us from identifying suitable endpoints that could reasonably be targeted. And furthermore, we all know that many mitochondrial disease patients undergo waxing and waning in symptoms, and being able to understand this is critically important, not only for endpoint selection, but also for selecting an appropriate duration for the clinical trial. Next, having biomarkers is critical for all stages of disease development, including early stage, preclinical development, as well as clinical development. In the preclinical setting, having suitable biomarkers allows identification of promising drug candidates, and it allows us to also establish early on reliable PK/PD models. In the clinical development, having biomarkers allows us to identify candidate responder populations, which can allow us to enrich clinical trials. It could also allow us to demonstrate drug target engagement in early stage development work, which can accelerate the overall development program. And finally, it also allows us to identify potential subgroups who are likely to respond to a drug, or allows us to better understand or identify different subgroups within a heterogeneous population. Next, objective metrics are better than composite scales. Rating scales that are used throughout pediatric disease and adult disease are often used by physicians to stage a disease, or at a single point in time, communicate how severe someone's symptoms are. Unfortunately, these scales are typically not developed to be used in clinical trials. They're used to be used maybe once to set a stage at one point in time, or say at some long interval of time, maybe annually or every other year, so we can follow someone's progression. That does not necessarily mean it's going to be useful in a clinical trial and can detect changes that might occur in the context of a shorter clinical trial. In addition, scales have an issue of inter-rater variability, which means that two different physicians may actually interpret a scale differently or may grade patients differently, which could introduce another level of variability in a clinical trial, which could impact the chances of success. Next is the issue of short trials leading to long delays. The rationale for conducting a short trial is clear. If it's a placebo-controlled trial, having a shorter trial may minimize the duration that a patient may be exposed to placebo. It also could lead to a shorter path of approval, and importantly, shorter trials tend to be less expensive. The entire drug development process is incredibly expensive, and obviously, the longer the clinical trial is, the higher its cost. So there's often an incentive for a company to do a shorter trial that would save money. The reality is that this short trial is not always the best plan. It's very hard to affect or demonstrate a clear clinical benefit in a heterogeneous disease in a short period of time. Often a longer period of time is needed to to account for some of that disease heterogeneity, or specifically a variable rate of disease progression. In addition, a longer clinical trial can account perhaps for the waxing and waning that occurs in many mitochondrial disease patients. Also, when you think about the need to do a subsequent clinical trial, if your first one was too short, well, what you've basically done now is lengthen the entire drug development process. It may have been a lot shorter process if a longer, appropriately designed trial was done the first time. Next is the importance of having an aggregated patient community. having an aggregated community where there's a clear culture of clinical trial participation really helps in recruiting patients for clinical trials, and also in having all patients, physicians, families, and other stakeholders really understand the trial process. It's often confusing for patients to understand that a clinical trial is not clinical care, that there may be specific inclusion criteria one needs to meet in order to participate in a trial. The need for a placebo phase can also be confusing to folks, as well as the need to travel to study sites. Many patients often ask, why can't I just go to my local doctor's office? Why do I have to travel hundreds of miles to be in a clinical trial? It's also important to have an aggregated community so that we can share important information about clinical trials and accelerate the enrollment process. Finally, all of these challenges are exacerbated by disease rarity. We can't overcome any of these individual challenges, such as trying to achieve homogeneity, by just enrolling hundreds more patients. They just simply don't exist. And these challenges also heightened by the fact that if patients need to travel to study sites, really ill patients may not be able to actually travel. And this may further limit the number of patients who can participate in a study, and may create a selection bias in that if only less severe patients participate in trials, we're excluding an important part of the community. And so this is a really important one to think about as we set up study sites and criteria for entry into a trial. So in conclusion, there's clearly a number of significant challenges to developing drugs for mitochondrial disease. And over the years, our understanding of these challenges has helped us improve our clinical development processes and increase our chances for success. I can say that over the past decade, there's been enormous strides made in our community in creating a culture of clinical trials and developing efforts that are coordinated across all stakeholders to address these gaps. These efforts are incredibly important and will continue to increase our chances for having successful drug development and delivering drug therapies for patients with mitochondrial disease. Thank you.
Okay, good morning, and thank you very much to UMDF for inviting me to talk about something very close to my heart for more than 20 years, which is the treatment of mitochondrial diseases. I'm Patrice Rioux. I started working 20 years ago at Repligen with Bob Nevio and Richard Haas and Bruce Cohen. And then I moved to California to start Edison Pharmaceuticals with Guy Miller. But after I joined Raptor Pharmaceuticals, I realized that I might have the real treatment for mitochondrial disease, a compound from the family of thiols, which is cysteine, N-acetylcysteine, glutathione, cysteamine, taurine, all these compounds with a sulfur in it. So let's look at this interesting slide, which shows one thing, which is that the cysteamine is the strongest chemical antioxidant, but its unique mechanism of action might actually rely on the fact that, as shown for the treatment of a rare disease called cystinosis, cysteamine binds cysteine to produce a disulfide that crosses any kind of biological barrier, including intestinal and blood-brain barrier, through the lysine transporter. In the cells, the so-called reduction of this disulfide leads to liberation of cysteine, and cysteine is the rate-limiting factor to build glutathione, which is the most potent biological antioxidant, but which is also poorly available, meaning direct supplementation of glutathione is not successful. So cysteamine has been tested in animal models of mitochondrial disease, but before that, it has been tested in multiple models of multiple indications, such as cystic fibrosis, neurodegenerative diseases, for example, Huntington's disease, Parkinson's disease, and also non-alcoholic steatohepatitis, sickle cell disease, malaria, and any kind of models. But only recently in animal models for mitochondrial diseases by Marnie Falk and her team at CHOP. So I actually remember going, visiting Marnie's site with the cattle of worms, the C. elegans, and the zebra fish aquariums at CHOP, where she tested some cysteamine provided by Raptor Pharmaceuticals when I was still working there. As can be seen on the slide, the models allow to explore several potential outcomes for treatment. I will not go through the results of these experimentation. Marnie can do that far better than me. However, a key point of these experimentation is that cysteamine was provided in continuous for this experimentation. Worms keep grazing in their plates, and fish keep drinking tasty water, kind of garlic taste. And that's where that's a key parameter here. That's a continuous, providing continuous flow of cysteamine flow or administration of cysteamine. The main conclusion of Marnie's publication was that cysteamine was active, but needed to be provided just enough and not too much. This is actually something well known since the initial publication by Jess Tenney about using cysteamine for the treatment of cystinosis, where the concentration in fibroblast assays has to be between 0.1 millimole and 1 millimole. Below, it doesn't work. Higher, it becomes toxic. And so this, we have to have in mind that we have to, it has actually, the maximum dose in cystinosis is 1.95 gram per meter square per day. And that's the upper limit, because if we go over that, it's actually, it could be toxic, we have seen in cystinosis. So let's talk about continuous exposure to antioxidant. As I said, we said continuous in the animal model. Cysteamine has been shown effective in, as I said, in multiple animal models for multiple indications. However, it's marketed since 1994 only for the treatment of cystinosis, despite all these models working where it's working, and not for any of the other indications. As seen for cystinosis, the reason is that you need to administer cysteamine in such a way you get 24/7 exposition to a minimal concentration in the blood, approximately 5 micromole. So that's something, how do we know this minimal concentration of 5 micromole? That's because in cystinosis, the breaking of cysteine in 2 molecules of cysteine and the incorporation of each cysteine into a disulfide cysteamine cysteine requires this minimal concentration. Otherwise, cysteine starts accumulating into crystals that ultimately destroy the cell. So to keep the 24/7 exposure, the high dose of immediate release formulation, cystagon, have to be administered strictly every 6 hours because of the very rapid elimination half-life. And as you see on the picture, after 6 hours of the immediate release, there is the impact or the exchange on cysteine doesn't work anymore. And so if you leave the patient without cysteamine after 6 hours, the cysteine goes up and it becomes toxic. So that's why there were several publications showing that actually you cannot do even 8 hours instead of 6 because it has an impact on the kidney function. When I was at Raptor, we developed a delayed release formulation of cysteamine, ProCysB, or at that time it was known as RP103, that can be given every 12 hours, but twice a day instead of once a day. That means that the same amount of daily dose, which means with the same amount of daily dose, means that a peak of concentration even bigger than cystagon with the immediate release, and therefore still with side effect, which most of these side effects are linked to this high peak concentration. It's linked to the local administration in the stomach, but also to the peak. We don't have the stomach effect with the delayed release, but we still have the peak effect. So a few years after leaving Raptor, and with a colleague of mine, and I patented a new cysteamine prodrug that has 2 main features. a low peak, as you can see here, and a longer exposure. So you see that with the normal cysteamine, the absorption is such that you have very rapidly a peak going up to 60 micromole, for example, with the cysteamine hydrochloride. But when you give our compound, our cysteamine prodrug, the peak is never higher than 40 micromole here. And the good thing is that even if when you increase the dose, you can see that you increase the duration of exposure without increasing the peak. So if you don't have the peak, you don't have the side effects. And if you have the longer exposure, you can give the drug every 12 hours or maybe even every or once a day. So when I was at Raptor, we decided to take advantage of the improved pharmacokinetics of RP103 versus the cystagon, the initial one, to initiate a clinical trial in mitochondrial diseases. And so we initiated the open label dose escalating study assessing safety, tolerability, efficacy of RP103 in mitochondrial disease, which is a MITO 001. That was cysteamine bitartrate delayed release capsules. The ProCysB were administered twice daily following the dose escalation design with a progressive weekly dose increase over the first 6 weeks. The starting dose was 0.2 gram per meter square per day, up to a maximum dose of 1 gram per meter square per day. And that's because we didn't know much. That was the first time cysteamine was given in patients with mitochondrial disease. So we wanted to actually keep the trial safe. I would say now that I would go a little bit higher than that. We know that in patients with cystinosis, or actually we know that to have this exchange of cysteamine, cysteine, and so having cysteamine carrying the cysteine for the building of glutathione, we need to be closer in between 1.3 and 1.9. So this trial, we had a very great list of investigators, Bruce Cohen, Fernando Salia, Richard Haas, Greg Hens, and David Vishkoshil. So these investigators recruited a significant number of patients, but I couldn't wait to see the results since I decided to leave Raptor after the board removed the initial CEO and replaced him with a new CEO who was not interested in developing RP103. That's why you have not seen RP103 in any other indication after I left, actually, and they did that because they wanted to keep the price for RP103 for ProCysB in cystinosis, knowing that the previous formulation, cystagon, cost approximately $10,000 a year. The ProCysB cost is approximately $250,000, so 25 times more a year. That's a good reason for them to stay in cystinosis. So when I designed the protocol, we had no a priori on the categories of patients who would respond to treatment. And therefore, we were open to enroll multiple sub-indications. Interestingly, I would say, as we have seen with our cysteamine prodrug now, the European Medicine Agency, for example, grants orphan designation to each subcategory separately and not for mitochondrial disease as a whole, which means that nowadays we'll have to do a clinical trial in Midas, one in Lee, one in Paul-G, and so on. So, to my knowledge, the results of this trial have not been published yet, and therefore, what I have here is what has been entered on the official website of clinicaltrial.gov. Due to the small number of patients in each subcategory, it's not surprising not to find anything statistically significant. However, it can be noted that here that the third, domain 3 actually grow, mostly reflecting the growth, shows some trend. That's a p-value of 0.08. As you can imagine, on 36 patients with very different subcategories, it's actually interesting to see that. And this trend can actually be seen even better on one of the secondary endpoints, which is change from baseline in gross motor function which is linked to the domain 3 of the global scale. You can see that it goes from 0.4 to 0.9 and then up to 4, 4.4, 2.2 at the last week. That's something to discuss actually with the main investigators. And following this trial, there was actually an extension, a long-term extension to assess cysteamine, to assess RP103 in children with inherited mitochondrial disease. And what is interesting is that 22 patients of the 36 received cysteamine bitartrate. And this trial was there to make sure patients, generally what we do when we do an extension like that, when we plan to do an extension like that, is that first patients, if the treatment is working for at least for some patients, they can benefit of it. And so the enrollment is easier because you're not going to tell patients that you're going to stop after X number of weeks and that's it and they're done. If it works, we have to be able to provide the drug for a longer period of time. Unfortunately, as you know, that stopped and that's it. So in conclusion, despite the lack of statistically significant results with RP103, I believe a cysteamine prodrug with a better pharmacokinetics than previous formulation should be tested in mitochondrial diseases, at least in some subcategories, which have to be determined by the investigators of the initial trial. Thank you very much for your attention.
All right, thanks very much. Those were 4 really interesting presentations covering clinical trials that have taken place for mitochondrial disease. We're back now with our panel, and Dr. Karaa is going to lead us through a moderated Q&A session.
Thank you, Phil. Thank you for all the speakers. I wanted to make sure that our community understands that it takes a village really to conduct these clinical trials. So really, I commend you and thank you for sharing all that wealth of information. I think what I came out with hearing all of your experiences is that we have you guys have helped us maybe establish a path forward. What I heard was that natural history studies are much needed, that endpoints are very important, and that maybe we need to think about longer periods of trial design when it comes to our heterogeneous primary mitochondrial disease population. And so if you had to do it all over again, what would be the one thing that you would focus the most on? We have a lot of new companies joining us and will be telling us about their projects and proposal for engaging the community in mitochondrial disease trials. So what would be your one advice to them? We'll start with Jim.
Thanks, Amel. Clearly, we just added too much heterogeneity to the trial. So obviously, hindsight's 20/20. But I guess if I were to do it all over again, I would focus on the nuclear DNA group where we saw the most benefit.
So what you're saying is that really knowing what your drug does before you design your trial is really important. So it might be more helpful to spend that a little bit more money to try to get that down very clearly before the trial starts. How about Colin?
Yeah, this is Melanie.
Actually, Melanie, because Colin is not here. Sorry, Melanie.
No worries. Particularly with our motor trial, the endpoints, I think, were a key factor in the design of that trial. So I think that's probably the biggest consideration moving forward. Our primary endpoint in the motor study was peak work based on the exercise testing, and implementing that across sites from a feasibility standpoint was challenging. So identifying endpoints that are feasible across multiple sites while minimizing variability, as many of the presentations have highlighted, is a key consideration, I think, moving forward.
Yeah, and that's a great feedback for us in the community. We need to do a better job at designing and validating outcome measures and endpoints that are unique to our patient population and heterogeneity. Okay, Matt?
I won't repeat Jim and Melanie's because they're both spot on. I would just emphasize there's no shortcut. And to really in planning a trial, take the time to actually talk to us, talk to people who've actually been in the trenches, and you know, pioneers take arrows, so learn from our arrows.
So am I hearing that you guys would be open to share your experiences with any newcomers in a more transparent and open forum?
Well, I've said to many folks over the years that, you know, in our early days when we had done some of these first trials, it amazed me that people didn't pick up the phone and call me. I gladly share my experiences because we're all in this together. And I think there's no lecture that's of appropriate length that would allow us to really get in depth on all of the learnings. And I think as, you know, you're looking at 4 people here who put their, you know, as a lot of people who put so much of their life into this, and there's so much that we've learned and see from where we're at that, you know, you may not even talk about publicly, but it's really important. And I would talk about it on a phone call because these are lessons people really need to learn if they want to succeed.
Thank you. That's really helpful. Patrice?
You know, as I said, actually, and that's something requested by the EMA now, that you have to design your trial for one specific indication. They ask us actually to apply for orphan designation for MILAS, and that's it. So clearly, yes, the heterogeneity is a problem. And something which is, and I appreciate one of the presentation earlier this morning, the fact that, and I did that for another disease with another compound, to actually, the FDA is now really interested in using the comparison with natural history of the disease, with placebo group. So clearly being far more open than the usual standard design of versus placebo, and show something. So that's good.
So I wanted to put it out there that as you have heard throughout the day today, data points and information about our patient is really important. And so any data point that comes from clinical trial is really precious. And I know that some of these trials have already been published. Would we get a commitment from you guys to publish even the negative outcome from the trials that have gone through? How can we make that happen as a community so we can understand and learn from prior trials, whether they have failed or became successful? Jim?
Absolutely, Mel. As you and I would discuss, we are committed to publish the results.
Very good. Melanie?
Yes, in fact, the results of the motor trial have been published. So yes, we're very accustomed to doing that.
And Matt?
Yeah, I think now clinicaltrials.gov requires you to put everything online. We're all required to do that. That was a rule that was put into place effective from a couple of years ago. And so obviously we're all committed to complying with rules and regulations.
And Patrice, I know you're not at Raptor anymore, and it's not Raptor anymore, but do you have any insight into whether this is ever going to be published?
No, I don't think so, because as I said, they are not interested in using this compound in mitochondrial disease. So maybe Greg Hens has already put a note in some publication about that trial, but nothing much. And the main issue is that effectively I cannot say, you know, it didn't work in this subcategory of patients, it worked better in the other one or something. Yeah, that's something I got from talking to Fernando or Greg or Bruce, and that's why they would be interested to going forward with this compound, but I don't know why. And I agree with you, that would be nice to understand why actually it didn't work in this category and more in the other, potentially more in the other one or something. No, I agree.
And Phil, do we have any questions from the chat, or should we?
Nothing specific, and of course the purpose of this panel isn't to talk about any specific intervention. I think in general, though, we've heard some really consistent themes right across all of the responses here. First, there's a consistent willingness to have discussions with others that are coming into the space and share those lessons learned. There's a willingness to put the effort into looking at data that's collected and realize how precious it is in a rare disease like mitochondrial disease, find a way to build upon that for future research. These are the aspects that I think are what's going to take us from the lessons learned category to the how do we do things better going forward. So I know I very much appreciate all the insights that were shared here today. If there's nothing else, Amel, did you want to cover anything else here?
Yeah, and you guys might not be able to answer this live now, but this is something that we hear from the community and that I wanted to make sure you heard, is that our patient, it takes a toll on our patient to be in these trials. And we really want to be an advocate for our patients as well, because we ask a lot of them to be in a trial, and it becomes a very emotional journey to be in a trial that eventually fails, or it takes much longer to get that approval. So for those trials that have gone through and have seemingly failed, can you tell the community what your larger plans are? Is this failed forever? Is there maybe a glimmer of hope that this can be looked at in more details, and we can maybe reinitiate trials, doing them in a different way, learning from of the shortcomings from the prior ones? I'm not going to be calling on names because I don't want to put you on the spot, but if you want to say something, please do.
Now, let me just say that I think you've got 4 people here who come from all with companies that are fully committed to developing drugs for mitochondrial disease. I think we're, to a person, we're all in this space. We've all been involved with companies that have been involved in developing drugs for mitochondrial disease for a long time. We're incredibly appreciative and understanding of the tremendous sacrifices and strains that clinical trials place on patients. And really, one thing I've learned is that this is a team effort. We as a community, whether it be industry, whether it be you as a clinician, whether it be Phil and his role at UMDF, and most importantly, the patients, we have to be in this together and work together and understand each other's role in this process. But I think you have here people who are 100% committed, and companies that are absolutely committed to developing therapies for people with mitochondrial disease.
Okay.
I second what Matt said.
Perfect.
Yes.
I couldn't say any better than him. I agree with what he said.
Yeah,
Well,
thanks,
thank
Matt.
you very much. Oh, Patrice, did you have something to say?
No. I say thanks, Matt. Yeah, he's good.
Well, thank you very much, everyone. This was very informative. And with that, we'll take a break and then we'll resume in a few minutes. Bye.
Good.
Thanks, everyone.
Thank you.
Thank you.